So I'm going to look at some factors that affected plant genetic transformation. So, as I've mentioned, you need a trait that's encoded by a single gene, ideally. You need a means of driving expression of the gene in plant cells. So I've already talked about organ-specific promoters. So you need to promote gene expression, and you need terminators as well. I'll be talking about this much more later. You need a means of putting the gene into a cell. So I've already talked about biolistics. And you need a means for selecting for transformants. Typically, you would be transforming one cell in your target tissues out of possibly several million. So you need a means for actually selecting this one transformed cell from the million non-transformed cells. And finally, you need a means of getting a whole plant back from a single transformed cell, or regeneration. Now I've put this at the end because when you're doing the protocol, this is the sort of order that they would actually happen. But when you look at plant transformation protocols, you'll find that they're all built around a means of getting a whole plant back from the single transformed cell. Unless you can get a whole plant back from a single transformed cell, there's no point in doing the protocol in the first place. So, useful single gene traits have been introduced into plants. Herbicide resistance is a big one. Normally glyphosate resistance, which is probably about 90% of all herbicide resistance genes that have been put in. Insect resistance, again, single gene trait, crystal toxin from Bacillus thuringiensis is the major one. But there are others for both herbicide resistance and insect resistance. Virus resistance, I mentioned you can put in coat protein genes. Seed protection, you can put in a whole load of toxic compounds like lectins, and these will protect the seeds from insects that would devour them during storage. There are a number of different ways of putting fungal resistance in. And finally, this delayed ripening. The first commercially available transgenic plant, the flavor saver tomato, was actually engineered with delayed ripening. So we're putting in ansense to polyglacturinase that actually breaks down the middle lamella of cells when they're ripening. Cold frost resistance. In my own laboratory we've engineered overexpression of proline in plants through a single transgene, and from that been able to get cold frost and drought resistance. But there are a number of different ways of getting cold frost and drought resistance into plants. Overexpression of proline is just one of them. High starch potatoes, if you're frying up potatoes for making chips, then the higher the starch content, the better the chips. Oil production is another one. I've already mentioned these biodegradable plastics or renewable plastics through polyhydroxybutyrate. Digestibility proteins, I've already mentioned antibodies. If we look at genes for pest resistance, there are genes for insect resistance and fungal resistance. The big one of these is the second one down, the Bacillus thuringiensis crystal toxin protein. But you can put in things like protease inhibitors, you can put in lectins, and you can put in things called RIPs, ribosome inactivating proteins. Now, some of these are toxic to humans as well. So for example, lectins, ribosome inactivating proteins, can be toxic to humans as well. But the great thing about food is that the vast majority of foods that we eat are cooked. In the cooking process, you denature the proteins. So all these compounds are actually, all these proteins are inactivated on cooking. And this is fair enough because there are some plants which we regularly eat, for example kidney beans, kidney beans contain a very, very poisonous lectin, and you can get serious food poisoning by eating raw kidney beans or improperly cooked kidney beans. But it's not normally a problem because we cook food, most of our food. For fungal resistance, you can introduce chitinases and beta-glucanases that will break down the cell wall of the fungus. The true fungi contain chitin and beta-glucan in their cell wall. So it's possible to use this approach for targeting the true fungi. One of the problems with this approach is that there are some fungus-like protists, for example the water molds, that don't have chitin and beta-glucan in their cell wall, they have cellulose instead. So for example Phytophthora infestans, the causal agent of the Irish potato famines, is not a true fungus, it's a water mold. Its cell wall is cellulose. So you cannot engineer resistance to Phytophthora infestans by introducing chitinases and beta-glucanases. Improved post-harvest properties is a potentially important method of plant genetic transformation. Up to 50% of harvested food is lost post-harvest in Africa. And as I've mentioned, any poisonous protein can be detoxified by heating and rendered safe in a food. Ripening control has been introduced into plants, as I said, by flavour saver tomatoes. But you can also include things like wheat germ agglutinin, which is another lectin, or cowpea trypsin inhibitor. Other useful traits you can put in are improved agronomic properties, like high starch potatoes, improved plant breeding. So ideally, in plant production, well, for example in maize, a lot of the crop plants that we actually harvest don't breed true. They are first generation hybrids. So in order to get that first generation hybrid, you have to have crossing between two different plants, and you have to prevent self-pollination. And one way of preventing self-pollination is by using pollen abortion. So you can do this. I've already mentioned cytoplasmic male sterility, which is mitochondrially encoded and therefore is not pollen transmissible. But it doesn't always work. So you can engineer in pollen-specific promoter plus RNAs, and the RNAs just digests all of the RNA in the cell, and you end up with pollen abortion. And to stop it killing all the cells in the plant, you put it behind a pollen-specific promoter. And finally, improve nutritional properties. So golden rice has been produced by Ingo Petricus and his group over a period of about 15 years, where you get increased levels of provitamin A. And this can lead to, so basically you can supplement your diet not with rice but with golden rice, and this will provide you with all the provitamin A that you need in order to produce vitamin A to prevent things like blindness